Hello everyone, today we are to talk about exam and specimens. Of course, there are a lot of them and it is very difficult not to confuse them one with another. And that's why I want to highlight at the beginning, this video is not to study specimens. You should study them apart, use our website. There is an atlas with many images in it, and also in all our lectures. There is an explanation about every specimen one by one, but this video is just to help you not to confuse specimens, how to distinguish them, and just use this video after you prepare specimens with the help of website and lectures. Okay, let's get started. First of all, I put specimens very distinctive, so it is very difficult to confuse them with others. Okay, spermatozoa, I think they very unique, and you can distinguish them because they are stained with the iron hematoxylin, shades of gray, and they have tails and head with acrosome, difficult to confuse. So let's go further, and here we have the specimens of the white adipose tissue, and adipocytes. These are large cells filled with lipids. Sometimes blood vessels could be found, but doesn't matter. This is a specimen form-like, and I think it is very unique, and it is difficult to confuse it with others. So if you know it, you will recognize it in the exam, I think. And then also pigment cells, also unique specimen, and you can see him here. They are brown in color and star-shaped. Think very unlike others. Okay, they are brown in color always, so even by the color you can distinguish them from all the other specimens. Then this is the specimen of loose connective tissue and areolar loose, and here it is stained with the iron hematoxylin, shades of gray. So loose connective tissue, difficult to confuse with other specimens, just color is very helpful. And also, of course, you can see here many blood vessels and elastic fibers forming this interconnected network, but doesn't matter. I think the specimen is very unique. Okay, then this specimen is all about hemomicrocirculatory bed. So here we can see arterioles, capillaries, different venules, and also I think difficult to confuse. You have just to know. Then specimens from embryology. Here we can see somite stage of the embryo. Here we can see the late gastrula and amniotic folds are present, forming the amnion, and this is the primitive streak. Primitive streak, I think this specimen is difficult to recognize because it has no distinctive features, just a line, but if you find this invagination in the center of the line, then this is the feature of the primitive streak, so you have to recognize it. Okay, then this specimen is about the internal ear and cochlea. Cochlea with organ of Corti inside, and I think also it is difficult to confuse with any other specimens. You remember scala media, scala vestibulis, scala tympani, and everything's very unlike other specimens. Then this is the endochondral ossification. Specimen is about formation of bone tissue, and this specimen is represented by the little bone made up of hyaline cartilage, and in the center, in the diaphysis, cartilage is already replaced with a bone. So here we can see woven or primary bone tissue forming bone trabeculum, interconnected oxyphilic in nature. So endochondral ossification, the specimen is represented by the small bone. Then another type of intramembranous ossification, or the formation of bone from the mesenchymal tissue. Here we can see also bone trabecula, and they are formed from the mesenchymal tissue. So here is the mesenchymal cells and bone trabecula with large giant osteoclast on the surface, here multinucleated, and osteoblast cells producing the bone matrix. If you look macroscopically, Then you can see that also the early stage of tooth formation, but this is for stomatological students. Okay, so here, this is the essence of the specimen, bone trabeculum. Then about bones, also we have to distinguish lamellar bone, but this specimen is usually green and brown in color, very unique. Usually specimens are purple and pink, and just by the color you can distinguish lamellar bone tissue from all the other specimens, green or brown. Of course, you can see here osteons with Haversian channel inside with osteocytes, and that's the feature of bone tissue. After that, cartilages. In cartilage tissue, you have to distinguish hyaline cartilage from elastic. So in all types of cartilages, you can see special groups of cells, isogenous groups, on the background of extracellular matrix. But the difference is that in hyaline cartilage, isogenous groups, they are more rounded, while in elastic cartilages, they are elongated. And as for the extracellular matrix, in elastic cartilage, the matrix is forming a network. There are elastic fibers, network-like. While in hyaline cartilage, the extracellular matrix is homogeneous. It is more transparent, and no fibers could be found there. So if you can see fibers, if you distinguish fibers between the cells, this is elastic. If there are no fibers and clear spaces, then this is the hyaline. And elongated isogenous groups, this is elastic cartilage. Also the color purple and pink for the hyaline and usually elastic cartilage is red or brown, but here as you can see it is purple as well. Then next specimen, these are myelinated nerve fibers. Myelinated nerve fibers with the nodes of Ranvier. They could be sectioned longitudinally or the transverse section could be given. So here we can see myelin envelope in cross section, and here the same is sectioned longitudinally. They are stained with osmium, so they are of a special color, difficult to confuse with others. Okay, I'm sorry, there were some technical problems. Let's continue. So, specimens from the central nervous system, they have common feature. They are black and white, and I think it is very difficult, very easy to distinguish them one from another in general because spinal cord, it has its butterfly-shaped gray matter in the center, and it is surrounded by the white matter. Then the cerebellar cortex, it has its also characteristic feature with very branched white matter, which is called arbor vitae cerebelli, or the tree of life, and here we can see very deep invagination on the surface, and the cerebral cortex, and let's look how to distinguish cerebral cortex from cerebellar cortex. So in cerebellar cortex, we have Purkinje cells in the middle layer. They are very easy distinguishable in different magnifications. Even in low magnification, you can easily distinguish Purkinje cells. Here they are, these dots, bits. And if we are talking about the cerebral cortex with its 6 layers, the borders between them are not distinguishable. They are not exact like lines. Yeah. So in cerebellar cortex we can easily distinguish 1, 2 and 3 layers. Layers are very distinguishable, and Purkinje cells are very prominent. While in cerebral cortex of large hemispheres, we can see cells forming 6 layers without distinctive borders. And the most large giant Betz cells, giant pyramidal cells, they are not so prominent as Purkinje. So I think you'll easily distinguish cerebellar cortex from cerebral just with the help of Purkinje cells. Look how prominent they are and how small Betz cells comparing with them. Okay, let's move on, and now let's talk about muscle tissues. And first of all, you can confuse smooth muscle tissue with cardiac muscle tissue and Purkinje fibers, and connective tissue in tendon. Really, tendon looks like smooth muscles, but the difference is that in tendon, all collagen fibers are parallel one with another. All lines are parallel, while in smooth muscle tissue, smooth myocytes, they form bundles, and bundles are divided with the connective tissue, and you can see they are interwoven different directions, so they are cross-sectioned, sometimes longitudinally sectioned, and they form bundles. While in tendon, once again, in tendon, all collagen fibers are parallel along the same line, while in smooth muscle tissue, bundles are formed. Then, how to distinguish smooth muscle tissue from cardiac? In cardiac muscle tissue, we have Purkinje fibers just under the endocardium, and if you see those giant conductive cardiomyocytes or Purkinje fibers, here they are with pale cytoplasm, glycogen granules inside, so this is the Purkinje fiber. In the specimen with a smooth muscle tissue, usually this is the urinary bladder, you will see transitional epithelium on the surface. So if you look at the top, and if you can see this wavy shaped line, and in high magnification transitional epithelium, so this is the smooth muscle and this is the urinary bladder. Yeah, but if you look at the top of the specimen, if you can see here those giant palely stained cells, these are Purkinje fibers. Okay, so do not look at the bulk of the specimen. If you look here, muscle tissue you can see, and here also muscle tissue, you can distinguish them. But if you look at the top and seeing this transitional epithelium or Purkinje fibers, you can distinguish them easily. Okay, so let's move on. Now, skeletal and cardiac muscle tissues. First of all, they are stained with the iron hematoxylin, so they are black and white. By the color, you can guess that this is the muscle tissue, but the task is to distinguish skeletal from cardiac. And first of all, if you see cross-sectioned cells, I think that this is the skeletal muscle tissue feature, because usually in our specimens, okay, in cardiac muscle tissue, all cells are parallel, and in the section they are oriented along the same line. So if you can see some of them longitudinally sectioned, some of them sectioned across the long axis, then this should be skeletal. This is the cardiac. But what is more helpful is nuclei. If nuclei are at the periphery, this is the skeletal. Okay, in cardiac muscle tissue, you can see the position of the nucleus because there is no cross section. And of course, you look for these intercalated discs. If you see intercalated disc, then this is the cardiac muscle tissue. If without those intercalated discs, then skeletal. Okay, let's move on. And now specimens from the nervous system. These are neurons stained with a Nissl substance, with a Nissl dye for Nissl substance, and you can see bluish granules inside. Actually, if to be honest, this is the only one specimen stained with blue color. So if you see this blue colored specimen, then this is the substance of Nissl with multipolar neurons. And macroscopically, we can see this butterfly-shaped gray matter because this is the spinal cord, but it is stained with another dye. Okay, then and this is the spinal ganglion, and it is incorporated in the dorsal root. Here is the ventral root, and here is the spinal nerve. And if we magnify pseudo-unipolar neurons, here they are, and these are neurons, but pseudo-unipolar, and do not forget they are concentrated in the spinal ganglion. Okay, and now lymphoid organs, and we are approaching to the most complicated topic, because really they look like each other, because they usually have lymphoid follicles inside, those spherical aggregations of lymphocytes. could be found in the lymph node, as well as in the tonsil, as well as in the spleen, lymphoid follicles. Notice that thymus, here it is, has no follicles. Okay, so if you see a follicle, then this is probably not thymus. Then in the thymus, medulla is lightly stained while cortex is dark. And do not confuse, sometimes medulla forms these spherical structures, but they are light, while follicles are always dark in color. Okay, so if you see light spherical structure, these are not follicles. Okay, and in thymus, medulla is always lightly stained, while cortex is dark. And both of them are homogeneous. There are no vessels, prominent vessels, or some crypts, anything else. And then thymus also has a special shape of its lobules, and they are separated by the space, like empty spaces usually. Okay, now let's look at these three organs. Very difficult not to confuse. So in the lymph node, follicles are usually concentrated in the cortex at its perimeter, while in the center usually they are not found, only solitary. Okay, so medulla is more light, while in cortex it is dark and follicles are aggregated at the cortex. In the spleen, follicles are distributed uniformly. They are neither in the center nor in the periphery, uniformly, without any characteristic pattern. Okay. In the tonsil, follicles are aggregated along the crypt. So here is the crypt, the invagination of the epithelium, and by each side, follicles are located along this line. So if you can see this space, this invagination, this empty space line, then this is probably a tonsil. Okay, now let's look at them one by one. So in the lymph node, follicles aggregated at the periphery. In the medulla, there are medullary cords, this network of medullary cords. And between medullary cords, there are sinuses, medullary sinuses. And you remember that they are filled with a reticular tissue. Here it is. So lymph node is used as a specimen, as a lymph node, and reticular tissue as well. So if you see high magnification of its medulla, then this is reticular tissue in it. Okay, then thymus, we have already discussed. The cortex is dark, medulla is light. Both of them are homogeneous, without any follicles, without any prominent other structures. So it is very easy to distinguish it. Tonsil. Once again, there is a crypt in the center of the tonsil, and this space, this cavity, helps to distinguish tonsil from other lymphoid structures. So lymphoid follicles here are aggregated along this invagination. If you see this invagination, this is the tonsil, because no empty spaces we can find in spleen, lymph node, or anywhere else. As for the spleen, as I told you, lymphoid follicles here are distributed uniformly, randomly, and it could be differentiated by the capsule. In the spleen, on the surface, you can see pink, more or less thick, capsule containing smooth muscle elements, and this capsule sends trabecula inside them, and those trabecula are very prominent and pink in color. So if you can see this pink capsula with trabeculae, this is the spleen. And one more feature is that lymphoid follicles are accompanied by the central artery. So if you see the central artery inside lymphoid follicle, this is probably a spleen. Okay, let's move on. Now, epithelial tissues. Here you can see the specimen with a simple columnar epithelial tissue. Sometimes it is simple cuboidal, depending on the height of the cell. Here they are more columnar than cuboidal. And this is just the specimen, not of the organ, but of the tissue. So to distinguish simple columnar epithelial tissue, here we can see ciliated epithelial tissue, respiratory epithelium, stratified squamous, non-keratinized, and transitional. How to distinguish them, we'll discuss later. Vessels. How to distinguish artery of elastic type from artery of muscular type, and vein First of all, artery of elastic type. It is stained with brown or dark red color, and it is represented by many wavy shaped lines, elastic membranes. So many membranes are aggregated in the tunica media, and I think it is a very distinctive feature, and we can easily distinguish aorta or elastic artery from other vessels. As for the muscular arteries, in the tunica media here we can see nuclei of smooth muscle cells. So if you can see here those purple nuclei, this is probably not an elastic artery, because in elastic artery nuclei are not visible, because only elastic fibers could be found. Then in the muscular artery, what we can see, the intima is forming wavy shaped line, internal elastic membrane. So here it is internal elastic membrane, transparent in color, and here is the external elastic membrane. They are not stained because hematoxylin and eosin is used, and it is it doesn't stain elastic fibers. As for the veins, in veins we also can see smooth muscle cells in the tunica media, but notice that no elastic membranes, no transparent lines. And if in the muscular artery intima is wavy shaped, here it is smooth. Okay, so also of course some veins are blue in color comparing with arteries, but sometimes veins are also stained with also purple and pink. So do not look at the color. Pay attention to the presence of elastic fibers and wavy shaped intima, which is smooth in veins. Okay, for example, here another example is given. This is the artery of muscular type, muscular artery. You can see wavy shaped intima and elastic fibers here, and this is the vein. So in vein intima is smooth, and what is besides here valves could be found, not always, but in some veins you can find valves, and you can see the irregular space of the lumen, while in arteries it is rounded, here always round and open. In vein it is collapsed usually. Okay, then, and this is the specimen of the hair follicle. I think difficult to confuse and very distinct. If you know, you recognize. And this is the sebaceous gland, the example of the alveolar branched, simple alveolar branched gland with holocrine type of secretion. I think also very difficult to confuse, because usually sebaceous glands, they accompany hair follicles. So if you see the hair follicle, then sebaceous gland is there, and some sweat glands are here. Okay, also this is the stratovascular keratinized epithelial tissue, which could be found in the skin of the finger. And here you can see stratum lucidum. Sorry. Stratum lucidum is here, and this is the finger epithelium. Then this is the hypothalamus with its third ventricle in the middle and butterfly-shaped wings. These are the paraventricular nuclei, and close to the chiasma opticus, these are the supraoptic nuclei. Actually, this specimen usually we do not include in the exam, but you have to know. It is also difficult to confuse with any others, so if you know, you recognize. Hypophysis or pituitary gland. Some students, they look at our website and they find the name pituitary gland, and when I write hypophysis, they can't choose it. They say there is no correct answer when they look for the options. So please do not forget that some organs, they have different names, and you have to know all the synonyms. So pituitary gland and hypophysis, pineal gland and epiphysis. Okay, do not confuse. Then I think it is difficult to confuse. You can see the adenohypophysis, neurohypophysis, and the Radke's pouch in between them. So you just have to know the specimen. And now many confusing specimens, and let's pay attention. First of all, this is the fundus of the stomach, and this is the colon or large intestine. You see, they look like each other. They are very similar, but usually fundus of the stomach is stained with shades of red with Congo red, a special dye. So they are red, while colon is usually purple and pink. And what helps you to distinguish them more exactly is the cellular composition of their glands. In the stomach, glands are tubular and parallel, and you can see parietal cells, large red parietal cells inside. They prevail. While in the colon, inside crypts, empty mucous cells predominate. So empty cells versus red cells. This is the most distinctive feature, and if you want to be sure, look at cells inside crypts and glands. Of course, they look like each other. You see both of them are tubular, glands and crypts. Tubular, and they are oriented parallel. But if you look at the cell, you can make the difference. Then, very confusing specimens here. Duodenum and pyloric part of the stomach. What is the subject of confusion? Very deep stomach pits look like villi in the duodenum. And gastric glands, here these are the pyloric glands, look like Brunner's glands in the duodenum. So how to distinguish? Very easy. Very, very easy. The thickness of muscular envelope. Muscularis in the duodenum is relatively thin comparing with the mucosa and submucosa. While in the pyloric part of the stomach, muscularis is very, very thick, much thicker than the mucosa, because close to the sphincter region. So look at the muscles and you'll see the difference very exactly. Another difference is the cellular composition of gastric pits. Here they are represented by mucocytes, transparent cells, transparent, colorless, while villi are usually covered with basophilic or eosinophilic enterocytes. So colorless cells and chromophilic cells. That's the difference, another difference. Then, how to distinguish duodenum from the jejunum? The answer is in Brunner's glands. If you see those glands here in the submucosa, this is the duodenum probably, yeah? But if there are no glands in the submucosa, only crypts could be found, then for sure this is the jejunum. Okay, then pancreas and parotid gland. Digestive glands, and they both are represented by basophilic secretory cells. They produce proteins. That's why they are similar. But in pancreas, we can't see those striated ducts, which are very prominent in the parotid. So in the parotid gland, inside the lobules, striated ducts are very, very prominent. So if you see them, then for sure this is the parotid, not pancreas. If you can't find those striated ducts and you suspect this is the pancreas, then look for the islets of Langerhans. They are not very distinctive. For example, in this specimen we can find it, yeah, but they are palely stained transparent regions on the background of the basophilic exocrine portion. So if you can't find striated ducts, but you find these islets of Langerhans, this is for sure the pancreas. Then to another salivary glands, very confusing. This is the submandibular and sublingual. It is not very difficult to distinguish them from parotid, because parotid salivary gland has no mucocytes. All cells are basophilic. So if you see any transparent mucocytes, then for sure it's not parotid. But submandibular and sublingual, they're very, very similar. So if you see that mucous cells predominate, they prevail, then this is sublingual. If you see some solitary mucous cells, but serous cells, they prevail, then this is the submandibular. Okay, but of course they are very, very similar. And if you choose one field of view, usually by the only one field of view, it is difficult to distinguish them. Okay, let's move on. Now, parotid gland versus parathyroid. They are similar, but of course striated ducts are very characteristic for parotid, so you can distinguish it. And in the parathyroid, Usually connective tissue is very rich in adipose cells. So many adipose cells could be found, and the lobules are not very distinctive. They are infiltrated with connective tissue, and the border is not regular. Like here, for example, you can see the lobule, and lobules are very exact in their shape. They have very exact borders. Here, borders are not so exact because connective tissue infiltrates them. Okay, so this is the parathyroid gland. Yeah, besides, I forgot to say that in the parotid gland, also connective tissue very well developed between the lobules. Here it is pink. While in pancreas, usually connective tissue is almost absent. So lobules are separated by empty spaces. So it also works. Very useful criterion. Next pair of organs. This is the parotid gland and liver. Actually, it is very difficult to confuse, but some similarity is characteristic. So liver is homogeneous specimen. Almost nothing can be distinguished except central veins. So if you can see homogeneous background with only some empty spaces, these are central veins. So this is probably a liver. And also you can distinguish here some thin-like borders between lobules. They should be hexagonal in shape, but as connective tissue is very poorly developed, then usually here no connective tissue, and lobules are not very distinctive. Only central veins and sometimes triads. Okay, so in the liver, look for the central veins. That's a helpful criterion, and also a very slight, very thin line between the lobules. After that, adrenal gland and the liver. If to look at the zona fasciculata, it might be confused with the liver, but that's why you have to analyze the whole specimen, do not look only in the center. In the adrenal gland at the periphery, you can see zona glomerulosa, and it helps you to distinguish it from the liver. Okay, and also in the center, medulla is darkly stained. And then another pair of organs, kidney and adrenal gland. Might be confused because they are spherical both and almost the same in color intensity, but in the adrenal gland, again, you find zona glomerulosa just under the capsule. If you look at the kidney, there is no zona glomerulosa. The cortex is more or less uniform. Also in the adrenal gland, in the center, medulla much more intensely stained, while in the kidney, medulla is more lightly stained. Okay, and of course in the kidney you can find those corpuscles or glomeruli, many glomeruli in the cortex. So this is the feature, and that's how you can distinguish kidney from the liver, because in liver nothing can be distinguished except central veins. If you find glomeruli, then this is the kidney. Then a lens. Inside a lens you can find large bronchi, and do not confuse it with the trachea, because in other words this specimen is unique and can be confused with others. And if we look at the lung and lung alveoli, they can be confused with the mammary gland. But what helps you to distinguish them one from another? Alveoli of mammary gland, they are spherical and rounded, and in lung alveoli they have irregular shape, and usually they are interconnected one with another, forming a continuous network of empty spaces. Also in the mammary gland, connective tissue is very prominent between the lobules. Then another pair is thyroid and mammary gland. In thyroid we can see follicles. In mammary gland, secretory portions of acinar cells. And you see that lactating breast and thyroid are very similar, but in breast we can see the horizontal line separating lipids and proteins, while colloid is homogeneous. There is no line in the center. Okay, this is the first helpful criterion. And the second criterion is the presence of connective tissue in mammary gland. In the thyroid, it's not very prominent. So first of all, look, this horizontal line is very, very helpful. Sometimes it is not very prominent. You have to be very attentive to distinguish it, but it works. And also connective tissue. After that, mammary gland also could be confused with the epididymis. Epididymis is also represented by spherical ducts. Look like here, for example, yeah? But in thyroid and mammary gland, there is the oxophilic secretion and homogeneous. There are no cells inside. But in the epididymis, there are cells. You can see those small dots. These are nuclei of spermatozoa. So in the epididymis, spermatozoa inside, while in mammary gland and thyroid, there are homogeneous secretion. Then the epididymis shouldn't be confused with the testis. Testis has a more thick wall, and spermatozoa are usually fixed to the wall. While in the epididymis, spermatozoa are separated from the wall. There is a space between spermatozoa and epithelium. Here, the space. If you see the space, then this is the epididymis. Here, spermatozoa are fixed, so this is the testis. And notice the thickness of cell. The spermatogenic epithelium is much thicker than columnar epithelium in the epididymis. Besides, here another epididymis is shown, and it is very confusing with the testis. One might suspect that this is the spermatogenic epithelium, but no, we can see giant stereocilia, and spermatozoa are in the center. There is a distance between epithelial cells and spermatozoa. There is a distance. So this is the epididymis, not the testis. Then prostate. Prostate might be confused slightly with mammary gland, but it has a very unique, like a frozen water on the window, frozen landscape. And you can see that secretory portions are not spherical or rounded, but very branched and has a special appearance, very unique. And lobules are separated with the fibromuscular stroma of connective tissue. Also in prostate, usually except glands, you can find also the ductus ejaculatorius and the prostatic urethra. So if you can find those giant ducts, this is probably the prostate. After that, there is an ovary. Ovary might be confused maybe with thyroid or mammary gland, but I think that good students can distinguish oocytes inside and large cavity of mature follicles and many primordial follicles at the periphery. So follicles are different in size, in shape, and some of them do not contain fluid but filled with the cells. Okay, then there is a tongue and two types of tongue papilla. These are the foliate and these are the filiform. Filiform are covered with a keratinized epithelial tissue. They are sharp and projected backwards. Okay, and I think they are very unusual, very difficult to confuse. Then these are the tubular organs, very, very easy to confuse. That's why be attentive. These are the uterus, ureter, and esophagus and trachea. And what's the difference? First of all, the shape of the lumen in trachea, it is always rounded and smooth, while in all the other organs they are collapsed, so folds are formed. Trachea, it also has a special seam-like appearance because smooth muscle cells on the posterior wall, they contract after the death, and that's why C-shaped cartilage turns into sigma-shaped. So trachea can be distinguished easily. Then let's look at those another two pairs and try to distinguish them. First of all, uterus and the esophagus. In the uterus, usually it is given in the proliferative phase of the menstrual cycle, so glands are tubular, longitudinal, and oriented parallel one with another. While in the esophagus, glands are coiled and are located in the submucosa. So the first difference is in the glands. Tubular, parallel. This is the uterus, coiled, unparalleled, this is the esophagus. The second feature is the epithelial tissue. In the esophagus, even in the low magnification, you can see very, very thick, stratified squamous non-keratinized epithelial tissue. In uterus, it is simple columnar, so you can distinguish on this magnification. There is no thick layer, yeah, only simple columnar. So if you can see thick epithelial tissue, this is for sure not the uterus. Then esophagus and ureter. In the ureter, glands are almost absent, as you can see. It helps to distinguish it from the uterus. But really, they are very similar with the esophagus. You can see here thick epithelium and here also thick epithelium. So first of all, you look at the glands. Glands are present in the esophagus and they are very not characteristic for the ureter. And the second feature is the epithelial tissue, transitional. Here you can see rounded cells and stratified squamous in esophagus. So you can see squamous elongated nuclei at the top. So look at the top, spherical cells, spherical nuclei, transitional, smooth nuclei. This is squamous. This is the esophagus. Okay, then trachea. In trachea, as I told you, the surface of epithelial tissue is smooth. And if you see the cartilage, hyaline cartilage, then for sure this is the trachea, because in any other organs the cartilage is present. Then also you have to distinguish the posterior wall of the eye, but it is very unique in the presence of pigmented cells in the midline. So this is the choroid, and it contains a lot of melanocytes, so it is dark in color. So this black line helps to distinguish. Sometimes also cornea could be included, but we do not include it in the exam specimen. So let's keep it. And the last specimen is about the placenta. Placenta is very confusing organ because it has very low unique features. There are villi of different size and diameter. Inside villi there are blood vessels, and around villi, this villi, they bathe in the blood of maternal blood. And placenta, it has its fetal part with amnion on the surface. So if you can see this amnion line, this is the fetal part of the placenta. And if you can see placenta, then this should be the amnion. Okay. And what is important to distinguish is that sometimes villi are not surrounded by the blood. Sometimes blood is washed away. So do not rely on this criterion. Of course, placenta should be represented by blood and villi, but sometimes only villi. So if you can see many spherical structures with mesenchymal tissue inside and very prominent blood vessels, this for sure is placenta. Okay. Oh, yeah. I'm sorry, this is the last specimen, and thank you for the attention. I wish you good luck and to do your best during the exam. But first of all, I want you to be very good doctors and apply this knowledge in your clinical practice. Okay, if you have any questions, any problems with distinguishing specimens, please write down below in the comments. And first of all, I advise you again, first of all, please attend our website, go there, find the page with our atlas and prepare specimens with the help of atlas. And only after that, use this video, because here practical tips how to distinguish, not how to learn, yeah? And the detailed description of every specimen is given in every lecture. So here we just wanted to create a very short and brief video just to refresh the knowledge and to help you not to confuse. Okay, thank you for the attention and good luck one more time.